Hello friends, today we will learn about enzymology. What do you mean by enzymology? And we'll discuss enzymology in details in this video. So let's begin. Enzymology. Enzymes are biocatalysts, the catalyst of life. A catalyst, first we will know about catalyst. What do you mean by catalyst? A catalyst is defined as a substance that increases the velocity of or rate of a chemical reaction without itself undergoing any change in the overall process. Enzymes may be defined as biocatalysts synthesized by living cells. They are protein in nature, colloidal, and thermolabile in character and specific in their action. In recent years, certain non-protein enzymes have also been identified. First is nomenclature and classification. In the early days, the enzymes were given names by their discoveries in an arbitrary manner. For example, the names pepsin, trypsin, and chymotrypsin convey no information about the function of the enzyme or the nature of the substrate on which they act. Enzymes are sometimes considered as under broad categories, that is intracellular enzymes and extracellular enzymes. What do you mean by intracellular enzymes? That means these enzymes are functional within cells where they are synthesized, and extracellular enzymes, that means these enzymes are active outside the cell. All the digestive enzymes belong to extracellular enzymes. The International Union of Biochemistry, in short IUB, appointed an enzyme commission in 1961. This committee made a thorough study of the existing enzymes and devised some basic principles for the classification and nomenclature of enzymes. Since 1964, the IUB system of enzyme classification has been in force. Enzymes are divided into 6 major classes. Each class on its own represents the general type of reaction brought about the enzymes of that class. First is oxidoreductase. Oxidoreductases. Enzymes involved in oxidation reduction reaction comes under this category. Next is second is transferases. Enzymes that catalyze the transfer of functional group, that is transferases. Hydrolases. Third is hydrolases. Enzymes that bring about hydrolysis of various compounds, that comes under hydrolases. Fourth point, liases. Enzymes specialized in the addition or removal of water, ammonia and carbon dioxide, etc. comes under liases. Fifth point is isomerases. Enzymes involved in all the isomerization reaction comes under isomerases. Now come to sixth and last enzyme, that is ligases. Enzymes catalyzing the synthetic reactions where two molecules are joined together and produce and ATP is used. ATP is used. Yeah, comes under ligases. Each class in turn is subdivided into subclasses which are further divided. The 4-digit enzyme commission number is assigned to each enzyme representing the class. First digit, class means first digit, subclass, that is second digit, subclass, that is third digit, and the individual enzyme, that is fourth digit. Now let's know about the chemical nature of the enzyme. All the enzymes are invariably proteins in recent years. However, a few RNA molecules have been shown to function as enzymes. Each enzyme has its own tertiary structure and specific conformation, which is very essential for its catalytic activity. The functional unit of enzyme is known as holoenzyme, which is often made up of apoenzyme. The apoenzyme means the protein part, and the coenzyme, that is non-protein part. Non-protein organic part. Holoenzyme, that means active enzyme, gives rise to apoenzyme plus coenzyme. Apoenzyme means protein, that is the protein part, and coenzyme is the non-protein part. Now let's know about the factors affecting enzyme activity. The contact between the enzyme and substrate is the most essential prerequisite for enzyme. For enzyme activity. Now let's know about the enzyme reactions. First is concentration of enzyme. As the concentration of the enzyme is increased, the velocity of the reaction proportionately increases. In fact, this property of enzyme is made use in determining the activities of serum enzymes for diagnosis of diseases. Second, in concentration of substrate. Increase in substrate concentration gradually increases the velocity of the enzyme reaction within the limit ranges of substrate levels. A rectangular hyperbola is obtained when velocity is plotted against the substrate concentration. Three distinct phases of the reaction are observed. Now let's know about the enzyme kinetics and Km value. The enzyme E and substrate S combines with each other to form an unstable enzyme substrate complex for the formation of proteins of the product. Enzyme plus substrate in reverse reaction in presence of K1 and K2 gives rise to enzyme substrate complex, gives then enzyme substrate in presence of K3 gives rise to enzyme plus product. Here in this reaction, K1, K2 and K3 represents the velocity constants for the respective reactions and as indicated by arrows. Km, let's know about Km. Km means the Michaelis-Menten constant. Km is the Michaelis-Menten constant, and or we can say Briggs and Haldane constant is given by the formula Km equals to K2 plus K3 divided by K1. Again repeating, Km equals to K2 plus K3 divided by K1. So for this also, for velocity also, it is there is one reaction V equals to V max into substrate divided by Km plus substrate, where V equals to measured velocity, V max is the maximum velocity, S equals to substrate concentration, and Km is the Michaelis-Menten constant. Km, or the Michaelis-Menten constant, is defined as the substrate concentration to produce half maximum velocity in an enzyme catalyzed reaction. It indicates that half of the enzyme molecules are bound with the substrate molecules when the substrate concentration equals the Km value. Km value is a constant and a characteristic feature of a given enzyme. It is represented for the measuring of the strength of enzyme substrate complex. A low Km value indicates a strong affinity between enzyme and substrate, whereas a high Km value reflects a weak affinity between them. For majority of enzymes, the Km value are in the range of 10 to the power minus 5 to 10 to the power minus 2 moles. Lineweaver-Bork double reciprocal plot. Let's know about Lineweaver-Bork double reciprocal plot. For the determination of Km value, the substrate saturation curve is not very accurate since Vmax is approximately asymptotically, and by taking the reciprocals of the equation, a straight line graph representation is obtained. It is much easier to calculate the Km from the interception on the x-axis, which is minus 1 by Km, minus of whole 1 by Km. Further, the double reciprocal plot is used in understanding the effect of various inhibitions. Now let's know about the third point, that is effect of temperature. Velocity of an enzyme reaction increases with the increase in temperature up to a maximum and then declines. A bell-shaped curve is usually observed. The optimum temperature for most of the enzymes is between 40 degrees centigrade to 45 degrees centigrade. However, a few enzymes, example venom phosphokinases, muscle adenylate kinase, are active even at 100 degrees centigrade. In general, when the enzymes are exposed to a temperature above 50 degrees centigrade, denaturation leading to derangement in the native structure of the protein and active site are seen. Majority of the enzyme become inactive at high temperature, that means above 70 degrees centigrade. Now let's know about the effect of pH. Increase in the hydrogen ion concentration, that is pH, considerably influences the enzyme activity, and a bell-shaped curve is normally obtained. Each enzyme has an optimum pH at which the velocity is maximum. Most of the enzymes of higher organisms shown optimal activity around natural pH, that means 6 to 8. There are, however, many exceptions like pepsin, that is 1 to 2, acid phosphatase, that is 4 to 5 pH, and alkaline phosphatase, that is 10 to 11 for optimum pH. Now let's know about the fifth point, that is effect of product concentration. The accumulation of reaction products generally decreases the enzyme velocity. For certain enzymes, the products combine with the active site of enzyme and form a loose complex and thus inhibit the enzyme activity. In the living system, this type of inhibition is generally prevented by a quick removal of products formed. Now let's know about the effect of activators. Some of the enzymes require certain inorganic metallic cations like Mg2+, Mn2+, Zn2+, Ca2+, CO2+, Cu2+, Na+, K+, etc. for their optimum activity. Rarely, anions are also needed for enzyme activity. Example, chlorine ion, that is Cl- for amylase. Now let's know about active site. Enzymes are big in size compared to substrate, which are relatively smaller. Evidently, a small portion of the huge enzyme molecule is directly involved in the substrate binding and catalysis. The active site of, uh, of an enzyme is defined as the small region at which the substrate, that is S, binds and participates in the catalysis. Now let's know about the silent features of active site. The existence of active site is due to the tertiary structure of protein resulting in three-dimensional native conformation. The active site is made up of amino acids which are far from each other in the linear sequence of amino acids. Primary structure of protein. For instance, the enzyme lysozyme has 129 amino acids. The active site is formed by the contribution of amino acid residues number, that is 35, 52, 62, 63 and 101. Third point is the active site is not rigid in structure and shape. It is rather flexible to promote the specific substrate binding. Next point is fourth is generally the active site possesses a substrate binding site and a catalytic site. The latter is for the catalysis of the specific reaction. Of the 20 amino acids that could be present in a enzyme structure, only some of them are repeatedly found at the active site of various enzymes. These amino acids are serine, aspartate, histidine, cysteine, lysine, arginine, glutamate, tyrosine, etc. Among these amino acids, serine is the most frequently found. Sixth point is the substrate binds the enzymes at the active site to form enzyme substrate complex, and the product P is released after the catalysis and the enzyme is available for reuse. Now let's know about enzyme inhibition. Enzyme inhibitor is defined as a substrate which binds with the enzyme and brings about a decrease in catalytic activity of that enzyme. The inhibitor may be organic or inorganic in nature. There are two broad categories of enzyme inhibition. First is reversible inhibition, second is irreversible inhibition. First of all, we will discuss about reversible inhibition. The inhibitor binds non-covalently with enzyme, and the enzyme inhibition can be reversed if the inhibitor is removed. The reversible inhibition is further subdivided into competitive inhibition and non-competitive inhibition. Let's discuss about first competitive inhibition. The inhibitor I, in short I, which closely resembles the real substrate S, is regarded as a substrate analog. The inhibitor competes with the substrate and binds at the active site of the enzyme but does not undergo any catalysis. As long as the competitive inhibitor holds the active site, the enzyme is not available for the substrate to bind. The relative concentration of the substrate and the inhibitor and their respective affinity with the enzyme determines the degree of competitive inhibition. The inhibition could be overcome by a high substrate concentration. In competitive inhibition, Km value increases whereas Vmax remains unchanged. The enzyme succinate dehydrogenase is a classical example of competitive inhibition with succinic acid as its substrate. Malonic acid has structural similarity with succinic acid and compete with the substrate for binding at the active site of SDH. Now let's know about antimetabolites. These are the chemical compounds that block the metabolic reactions by the compounds by their inhibitory action on enzymes. Antimetabolites are usually structural analog of substrates and thus are competitive inhibitors. They are in use for the treatment of cancer, gout, etc. The term antivitamins is used for the antimetabolites which block the biochemical actions of vitamins causing deficiencies. Example: sulfanylamide, dicumarol. Now let's know about second inhibition, non-competitive inhibition. This inhibitor binds at a site other than the active site on the enzyme surface. This binding impairs the enzyme function. The inhibitor has no structural resemblance with the substrate. However, there are these there usually exists a strong affinity for the inhibitor to bind at the second site. In fact, the inhibitor does not interfere with the enzyme substrate binding, but the catalysis is prevented, possibly due to a dist- distortion in the enzyme conformation. The inhibitor generally binds with the enzymes as well as the enzyme substrate complex. For the non-competitive inhibition, the Km value is unchanged while Vmax is lowered. Heavy metal ions can non-competitively inhibit the enzymes by binding with cysteine sulfhydryl groups. Second is, let's know about second inhibition, irreversible inhibition. Irreversible and irreversible, it is the second one. The inhibitors bind covalently with the enzymes and inactivate them, which is irreversible. These inhibitors are usually toxic substances which may be present naturally.